My name is Jihan Lee. I'm CSO of the Protier Biotech. Protier Biotech developed the innovative therapeutics using the molecular glue, and we found in 2022. And recently, we closed the pre-A $2.4 million US, and the GSPT1 molecular glue and GSPT1 ADC payload and V1 molecular glue are our main pipelines. We have more other in the discovery stage. And the Protier glue platform and MARC platform is our core technology. And MARC platform, actually, we replaced the payload with our molecular glue. The company led by Hyun-Taek Kim, CEO, he has more than 25 years experience in the pharmaceutical area and structure-based drug development. There are more than 2,000 GSPT1 degrader patent, so it's hard to get a new one. And we got the new one. Our patents got the novelty and inventive step confirmed by the written opinion of the ISA. Traditional modalities, actually the inhibitor bind to the active site and then inhibit the protein function. And molecular glue modalities, molecular glue actually induce the binding of E3 ligase and the target protein and degrade the target protein. Molecular glues can solve the unmet needs. Molecular glue can target the undruggable targets and no active site needed, and it degrades the target, not inhibit, and it's a low molecular weight. So it's totally different from the PROTAC. Our GSPT1 target is the indication, the multiple myeloma first, and we're gonna expand to the solid tumor. It's oral and in vivo, good in vivo efficacy and high PK exposure. The competitors are the Monterosa MRT-2359. Multiple myeloma target will be $37 billion US by 2029. And bortezomib and lenalidomide and dexamethasone are the standard care treatment. 70 to 90% of patients relapse after their first treatment. So there is a new product, the drug needed in this field. Our compound, PTB6003, has a better degradation activity than the MRT-2359. MRT-2359 is phase 1, and we have a really good selectivity. Our compound also has a submicromolar anti-tumor activity in the a lot of cell lines, hematologic cancer and solid tumor. But we select the first indication as multiple myeloma. The multiple myeloma in vivo model, animal models, it's for 14 days QD and PO. We compare with the lenalidomide 10 mpk and Monterosa 1 mpk. Our compound is 1.3 and 0.1 mpk. And our compound 1 mpk actually the tumor regression, completely regression. And then Monterosa 2359 tumor stable. Our compound 0.3 mpk and the efficacy similar to the lenalidomide 10 mpk. Lenalidomide is a big blockbuster drugs. It's a standard of care treatment. And so our 0.3 mpk and the lenalidomide 10 mpk showed a similar activity. So our compound 33 times better than the lenalidomide. And but no body weight changes observed in this study. When you compare with the other GSPT1 degraders, we use most 1 mpk, the highest 1 mpk, and then BMS 30 and 40 mpk, and it's IP, it's not the oral. And then other competitors also use the really high dose. So ours is the very lowest dose with the PO. When you look at the pink color, the GSPT, the tumor, almost no tumor left, actually. And then the number 2 samples show the little tumor. So we look at the GSPT1 level in the sample 2 of the 1 MPK of our compound, and there is no GSPT1 level showed. So GSPT1 actually degraded in the tumor. This is most compelling data. This is real patient data, the new patient and the relapsed patient data. The new patient, actually we compare with the lenalidomide and mezigdomide. Lenalidomide, the blockbuster drug in the market right now, and the mezigdomide is developed by the BMS. BMS is saying that the mezigdomide is in phase 3, and mezigdomide is 100 times better than lenalidomide. And our compound is better than these 2 compounds, the lenalidomide and mezigdomide. So it is very compelling data. So in the clinical trial, we're gonna replace the bortezomib and lenalidomide with PTB 6003 later. We also have a MAC platform. We are using the PTB 6003 as the payload. We are making our own ADC. I'm gonna skip this. This is our business models. We're gonna create the revenue with the early license out before the phase 1. This is our business model. We have 2 platforms, molecular glue platforms and ADC, MAC platforms. Let me summarize our compound. PTB 6003 has a very potent target degradation and strong anti-tumor activity and good PK profile and very good potent in vivo efficacy at lowest dose. And mouse non-GLP tox study is ongoing. Thank you.
So you had said that your business plan is to license out your technology before phase 1.
Phase 1, yes.
So do you have people interested?
Actually, the J&J actually very interested in the multiple myeloma, and then I met the BD person last year, and then they said they need a little bit of a tox study, and then the proteomic study. So we are making right now. Proteomic is already done, and then we are doing the non-GLP tox mouse, and then the GLP tox mouse. We're gonna do that. And the non-GLP is ongoing, so they're interested in. And once we have that data, then they want to share that. And then we also, I think, first strategy priority for us right now, we are like to the research collaboration of the license out of the payload as a payload, and then one company in Switzerland actually call us, and then we had a meeting last month, and then they really interested in the as a payload because they're looking for the degrader payload, and there are not many molecular glue payload, PROTAC payload, yes, but not many the molecular glue payload, yes.
Right. Yeah, I just thought that with recent years, most people aren't interested in licensing early technologies.
I'm sorry?
Companies aren't interested in licensing very early candidates. They're waiting for later and later, even out to phase 2, phase 3. And so that's why I was just wondering if you actually had conversations where people would be interested in licensing it already. Yeah. So that's great.
Yes. Okay. Okay. Thank you.